Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at autosomal linkage. So let's just remind ourselves about a chromatogram. Now we know as humans that we have 23 pairs of chromosomes. If we look at the chromatogram, we can see that the longest chromosome is chromosome number 1, and we can work our way all the way down to chromosome 22, which is the shortest one, and then chromosome 23, which is our sex chromosomes. Now all our chromosomes come in pairs, and therefore our autosomal linkage is talking about our chromosomes from number 1 to number 22 because they have the same genes at the same loci at each of the chromosomes because they're the same length. So these are homologous chromosomes, and we talk about them being an autosomal linkage if we were to look at, for example, chromosome number 4. And as we're looking at chromosome number 4, we identify that there is gene A and there is gene B. Now, this individual is heterozygous for both gene A and for gene B. That means that they have the dominant allele for both A and B, as well as one copy of the recessive allele for A and B. Now, what's very different to what we know before when we talked about dihybrid crosses and how independent assortment of alleles can occur. When we have what's known as a linked gene, those linked genes are going to be inherited as one unit. So if that's the case, what that means is you would get capital A, capital B, or small a, small b. There is no independent assortment during meiosis. They are linked, they are one unit, therefore you inherit the capitals, the dominants together, or you inherit the recessives together in my example. Now there is a little caveat here which says unless the alleles are separated by chiasmata. So this is important for us to understand, so let's take a step back. We have our chromosomes. Our chromosomes have got gene A and gene B, and we know that they will go through, during S phase, DNA replication. So we have duplicated through the semi-conservative replication. We have duplicated to now have these two chromosomes, which are made up of sister chromatids. Now, when we get to prophase 1, which is the first part of our meiosis process, when we get to prophase 1, crossing over can occur. Now what that means is that there are points as they line up as homologous chromosomes, the same length with the same genes at the same location, they line up in bivalence, and sometimes they have what's called chiasms form, a place in which they will exchange genetic material. So that when we go through meiosis 2, when we pull those chromatids apart, what's happened is they have actually exchanged their information. So during meiosis 1, during anaphase 1 of meiosis 1, my homologous chromosomes are pulled apart. Well, because my homologous chromosomes are pulled apart, that genetic material will have switched. So during meiosis 2, during anaphase 2, my sister chromatids are pulled apart. And what we get is we end up with what's known as recombinants. These are chromosomes that are not the same as the original DNA. So if we have a recombinant offspring, they will have a different combination of alleles that have been inherited from their parents. And this was very evident when we looked at Drosophila. So if we understand that recombinants are different from the parental alleles that were passed on, we can actually work out what's known as our recombination frequency. What we'd have to do is we'd have to calculate the number of recombinant offspring and divide that by the total number of offspring. Now, what they love to do in this type of question is they love to throw in a statistical test. So I'm going to bring in chi-squared here because it comes in very clearly with linked genes. So when we do a chi-squared test, we are looking at carrying out a statistical test that's going to measure the size of the difference between the results that you have observed and those that were expected. And you should know a null hypothesis. So the null hypothesis is always that there is no significant difference between what we expect and what we observe. Therefore, we can say that any difference we do see is due to chance. Well, if you understand that chi-squared is a statistical test that you're going to use to measure that difference, you will be given this equation in the exam. So you should know that to calculate your chi-squared, you need to do the sum of the observed minus the expected squared divided by your expected number. So let's put that into practice with a question then. I have two heterozygous plants which are displaying purple flowers with long pollen, and we are going to cross those two heterozygous individuals. The results are as follow. We end up with 4,831 purple long, 390 purple round. Round must be the recessive trait because I've been told in the question that they're heterozygous and I didn't see round in that list above. So always double check the stem of the question for that. Then we have 393 red. Oh, look, there we go. There is our color again. So our recessive trait is red because we didn't have that with our heterozygous. It was being masked by the presence of a dominant allele. But we have 393 which are red and long and 1,338 which are red and round. So if it were to follow Mendel's law of independent assortment, we would expect the ratio to be 9:3 to 3:1. So therefore, what we need to do is we need to calculate what is our expected numbers. So what's our sample size? Well, our sample size is 6,952. Then what we need to do is we need to divide that by 16 to work out what 1 would be. So our recessive trait, we would expect 434.5. And then we just times that number by the 9 to give us our expected 9. And we times that number by our 3 to get our 3s. And then what we do is we input that information into our equation. So we have our chi-squared is the sum of the observed minus the expected squared divided by the expected. So as you can see, you put that into your calculator and what do you get? My chi-squared value is 3,771.5913. So what do I do with this now? What's the next step? Well, the next step is they're going to expect you to be able to say whether or not you are rejecting or accepting that null hypothesis. So we've done our calculation, we've worked out our chi-squared value, and now what we need to do is we need to see if this is significant or not. So the first thing we're going to do is we're going to work out our degrees of freedom. Now the degrees of freedom is always your sample size n minus 1. So there was 4 different categories that we had here. So 4 minus 1. So that gives us degrees of freedom of 3. So we go to 3 on our table and I have highlighted all the way along here, but we want to use what we do in biology, and that is a probability of 0.05. So this is where we always go, is 0.05 first. That's our part where what we're saying is we would be 95% confident that 5% was due to chance. So if we read the value, we can see that our critical value here is 7.82. I'm just reading that straight off the table. So I go to where my degrees of freedom lies, I go to the 0.05 probability, and where they meet is the number that I need to use. So what does that then mean? Well, what I then say is that my chi-squared value is significantly higher than that critical value of 7.82. Therefore, the difference is significant. Also, if you actually look at the probability of 0.01, I'm way higher than that as well. Therefore, I'm 99% certain that the results are not due to chance. Because these do not follow Mendel's law of independent assortment, the 9:3 to 3:1 ratio, I am rejecting that null hypothesis. I can therefore assume that potentially these two different traits, the color of the purple and the length of the pollen, is determined by linked genes. So therefore, one of the reasons that my ratios do not follow Mendel's law of independent assortment is because I have linked genes, autosomal linked genes. Now, I have done another video on another reason why it might not follow the 9:3 to 3:1 ratio. So if you haven't done so already, do check out my epistasis video. But I hope you've liked this video. If you have, please do click on the like button and subscribe to my channel. I would love to keep making more videos for you. Also, do check out my revision platform, www.aiqchat.com.